In this experiment, you're going to be learning a new technique known as titration, which is an experimental method for determining an unknown concentration. So there are lots of different contexts where this type of thing can be applied. But what titrations are generally used for is determining some kind of unknown concentration. So in this particular lab, you are going to be determining the concentration of acetic acid in vinegar. So acetic acid is the acidic component in most vinegars. We're using just a store-bought white vinegar. And you're going to be determining how much acid, how many moles of acid are present in each liter of vinegar. So what we do is we take a sample of the solution that we're trying to determine the concentration of, and we add something that it will react with until we've added just enough of that other reactant so that all of our original stuff is gone. So in this lab, you're going to be reacting the acetic acid in the solution with sodium hydroxide, which produces water and sodium acetate as products. So if we gradually add the sodium hydroxide into the solution until we've added just enough, as long as we know the molarity of the sodium hydroxide, we'll be able to figure out how many moles of acid were present in the vinegar, and as long as we know the volume, then we can figure out the concentration. I'll do a separate video with a walkthrough of the calculations for this, but that's the general gist. So how do we know when we've added just enough? That is going to depend on your reaction. For this one, we'll be using a pH indicator known as phenolphthalein that changes color once our solution becomes basic, which is when all of our acid has been consumed. So this will change color. In this case, this indicator changes from clear to pink when all of our acid has been consumed. So phenolphthalein is clear in acidic and neutral solutions and turns pink in basic solution. So once all of our acid is gone, our solution will become basic, which will cause our indicator to turn pink. So we will be using a new type of glassware to do this that allows us to gradually dispense the sodium hydroxide solution while keeping track of how much we've added. So this new piece of equipment is known as a burette. So it is used for measuring or used for dispensing one reactant known as our titrant in a titration. And using burettes is a little bit of a skill in a couple different ways. Burettes are measuring the amount that has been dispensed from them. How much sodium hydroxide have you let go into your vinegar solution? They do not measure how much they currently contain. So that's a little bit different than other types of glassware that you're used to. Different than, say, a graduated cylinder, even though this is a burette is a cylinder with graduations. It is different in that 0 is actually at the top, and the maximum capacity for ours, that's 50, that will vary, but the maximum capacity is at the bottom. So it's upside down from what you're usually used to reading, which you need to keep in mind when you're recording your initial and final burette readings in the experiment, which I'll be demonstrating in just a minute. So when we look up close at a burette, 0 is at the top. So you'll find small numbers above big numbers. So for instance, in this example here, you always want to read a burette at eye level. So if it's not at eye level, you want to move it on its stand, either take it down so you can read it, or move it up and down at least, so that the liquid level is right at your eye level. So we'll eyeball here, some creepy eyelashes. We want to put eye level right at the level of our liquid. And note that this is between 10 and 11. So this is 10 point something. The tendency is to think of it as being above 11 is what you're used to, so this must be 11 point something, but burettes are backwards. We're in between 10 and 11 here. So we have each of these little marks is 1/10 of a milliliter. So we have 10.1, 0.2, 0.3, 0.4, 0.5, 0.6. So this is in between 10.5 and 10.6. We always want to read the bottom of the meniscus, the bottom of the curve formed by our liquid. So I would call this probably about 10.57 or so. We always want to read to 2 decimal places. We can estimate 1 place further than is actually marked on our device. So we can clearly see that this is between 10.5 and 10.6, so we can estimate that extra decimal place. It's a little bit closer to 10.6 to me, so I'd call it about 10.57, but that last digit can vary. That last digit is an estimation, but you should always read burettes to 2 decimal places. So essentially what you're going to be doing in this experiment is setting up your burette, filling it with, well, it'll be pre-filled for you with sodium hydroxide. And our burette also has a valve down the bottom known as the stopcock, which has a little handle on it that you can open and close. And that's what starts and stops the flow of NaOH out of your burette. So as you open that, sodium hydroxide can flow, and you can control the rate at which it's allowed to come out of the end of the burette. You can open it all the way and let it come out pretty fast. Do not recommend generally for titrations, or you can open it kind of on the diagonal and get a dropwise flow that you can control more easily. You don't want to go that slowly throughout the entire titration, or it'll just take you a really long time. You could, but it would take you forever. But once you realize that you're getting close to this stopping point, and I'll explain that more in the demonstration portion, then you want to slow down and control the flow so that you can get, as soon as that color change happens, you can stop your titration and be finished. First, I'm going to demonstrate the setup of the equipment for this procedure. So in using a burette, it is hooked up to a ring stand to keep it upright. So we use one of these clamps to hold it to the ring stand. The center of the clamp goes over the pole, and then these little parts on the side are for holding burettes. So each of these clamps can hold 2 burettes. We're just going to be using one side. So I can put this over the pole, tighten it at a comfortable height, and then I can attach my burette. So again, we can see the numbers on the burette are lower at the top and they increase as you go down. So as a reminder, the burette is measuring how much has been dispensed from it, not how much it currently contains. So 0 is at the top and these go to 50 at the bottom. So I can clamp this to my stand. Before I fill it up, I want to make sure that my stopcock is closed. So this position here, where the handle is parallel to the tube, means it's open, which is how we store them. And then this position where the handle is perpendicular to the tube is closed, which means that no liquid will come out the bottom. So I want to make sure it's closed before I pour anything into it. And then I can clip this up onto my stand. So we need to clean this before we do an experiment with it, before we fill it up with our final solution. So we will start with a little bit of water, just to rinse out whatever may have been in here before. So 5 or 10 milliliters will usually do the trick to get it nice and clean. It should have been cleaned previously anyhow, but always good to give it a rinse so you know for yourself that it is clean. So then I can open my stopcock and let that water flow out. Again, this is just a rinse to get it nice and clean. And I can wait for that to drain completely. So the next thing that I'm gonna do is rinse it with just a little bit of the solution that I am ultimately gonna fill it with. So once I've rinsed it, it's now a little bit wet on the inside, and we don't want even that little bit of volume of water to mess with the concentration of this stuff in our burette. So by rinsing with a little bit of my sodium hydroxide solution, that's going to ensure that that extra water has been flushed out, and then I can fill it completely with the sodium hydroxide, knowing that the concentration of sodium hydroxide is what it says on the label. So I can do that now that my water has finished draining. Just pour a little bit of the sodium hydroxide in, and then once again, I can open my valve here and let that drain completely through the bottom. Once that's finished, I can fill it up completely with the sodium hydroxide, and my burette is ready to go. All right, that is drained, so I'm closing that again to fill it up all the way to the top. So I usually fill it actually a little bit above the 0 line, because there is one more thing that we have to do, and that is to open the valve one last time just to let the air bubble out. So right now, because I let this drain completely with my last sample, you can see that there's an air bubble in here. So there's liquid down the bottom, but then this area is air, and we don't want that making the volume dispensed, or we don't want that included in the volume dispensed since it's not part of our solution. So by opening my valve one last time, I can flush that bubble out, and now I'm ready to go. I would want to write down my initial burette reading. It's not quite at 0, but that's perfectly fine. Mine's somewhere between 0 and 1-ish. It doesn't matter as long as you record exactly what it is. Now we're going to prepare a sample for titration. So in this lab, you're trying to determine the concentration of acid in vinegar. Acetic acid is the primary acid present in vinegars. So you'll be using white vinegar that we want to take a sample of. So 8 to 10 milliliters is what you're looking for. Anywhere in that range is fine. So we can use a graduated cylinder to measure out our vinegar. And add that to a small Erlenmeyer flask. Now we want to make sure that all of the acid has completely transferred from here into here. I actually skipped a step. Don't do this in your lab. You want to record the exact volume of vinegar that you're using. So before you pour it into your flask, record to 2 decimal places the exact volume, estimating the second decimal place, and then add it to your reaction flask. To ensure that all of our acid has actually transferred, we want to rinse out this graduated cylinder and add this water to our flask. The volume of the water doesn't matter. As long as you know the volume of the vinegar, you're good to go. So I would do 2 rinses, fill the graduated cylinder up, pretty much all the way and add that water into your reaction flask. So the one other thing we need to add is our pH indicator, which is phenolphthalein. So this is what's going to tell us when to stop our titration. This will change color at a pH that is right when all of your acid will have been used up. So phenolphthalein is clear in acidic and neutral solutions, and it is pink in a basic solution. So we want to add just a few drops, like 2 or 3 drops from your tiny little indicator bottle here to your sample solution, and that is good to go. So at this point it should be clear. If it turns pink right away, something is wrong. Your flask was maybe dirty, but you'll have to dump it all out and start again. So it should stay clear for now. But during your titration, you will start to see little flashes of pink, and eventually the pink color will persist. And that first sign of pink that doesn't go away is when you stop, it's when you are done. So when you're getting close to that is where you want to slow down the addition of the base into your flask to try and get, down to 1 or 2 drops, knowing when that pink change actually happens. You want the palest pink color you can. The paler the better. If you have to ask, is this pink enough? It's probably perfect. The answer is yes. If it's pink enough. If you get too hot magenta, you have gone too far. Not a big deal for this lab, but just be aware that paler is better here. So now we are ready to titrate. So we've got our reaction flask underneath our burette. Our burette is filled. We've recorded our initial burette reading. So once we open this, sodium hydroxide will begin to flow into my flask, and we should start to see little flashes of pink that then quickly go away. If you don't start to see a little bit of pink pretty soon, you probably forgot to add the indicator. If you do that soon enough, you can save your trial. If you add the indicator and the whole thing turns pink, you've gone too far and you'll have to start over. But if you add the indicator and it stays clear, you can just continue titrating, no big deal. So as we go through here, you'll start to notice that the pink lingers longer and longer. And that is when you want to slow it down. So you'll notice that I've kind of been adding it in bursts so far. But once I get to the point where the pink is sticking around for a few seconds, what I want to do is really, really slowly open my valve here so that I get it at this nice drop-wise sort of rate where it's adding 1 drop at a time at a reasonable pace that I can stop it in between drops when I want to. I'm getting pretty close since the pink is sticking around for a while before it disappears, which means that in probably just a few more drops, I stopped it for a moment there since the pink was lasting so long. At this point, I want to try and add it really just 1 drop at a time. So if I open it just far enough to let a single drop out and let that dissipate, I can repeat that process however many times I need to get to my endpoint. So we can see that 1 drop took us from clear to actually this pretty bright pink color. But what we're looking for is for it to be as pale as possible. I couldn't have done any better than adding a single drop. Sometimes there's some luck of the draw involved in getting a pale pink versus a not so pale pink. But the burette allows us to have really fine control over the volume. So at this point, we would want to record our final burette reading, and we can dump this solution down the drain and repeat this procedure for a second trial, making sure to rinse your flask in between.